Good afternoon, everyone. Are there any questions so far about anything, any science, any mechanics in the course? Any time you have a question, even during the time I'm talking, you're welcome to ask it. I don't mind, okay? Don't have to wait till later. Yes, sorry.
Not about the material, just about, I know our test is in like 2.5 weeks.
Right.
So what do you recommend doing?
What do I recommend doing?
So is it mostly like lecture-based? What's the format?
So the format of the test will be short answers. I'll put up last year's test so you can see the format. It'll be different. I'll give you some study questions beforehand. We'll cover what's in the lectures. Nothing outside of the lectures. I won't ask you anything that's in the book that I haven't covered, for example. All right? And I try to emphasize on the quizzes every week what I think are the important points. So those will be the focus of the exam, among other things as well. So I think if you just concentrate on what we do here, if you can listen to the lecture a few times, it really helps to reinforce it, because once is often not sufficient. But you'll know what I want you to learn. And like today we have some slides which show exactly what I want you to get away from this. Okay? Anything else? Yes? It depends when I put them up. So I didn't put the ones up till last night. So you get a week each time, basically. It's a week. Yeah, and if you don't make it for whatever reason, just tell me and I'll open them up again. Okay? Anything else? It's really, my goal in this course is to have you learn virology, okay? I think that's the most important thing. Unfortunately, I have to give you a grade, so I have to figure out a way to do that, but I want you to enjoy it and get the most out of it. Just the good thing is when the course is over, it'll be warm, right? Yes? I think when you start it, there's a little red number in the upper left that says you have until. So I just started taking one, and it said, yeah, I have till 6 p.m. So I give you a couple of hours. If you can't finish, I mean, today's quiz is only 3 questions, so it's not a problem. Each one will be 2 lectures, so it's not going to be any more than 6 or 7 questions. So you have plenty of time to do it. But if you have a problem, you know, and you want to redo it, just tell me, because I want you to use it as a learning tool. Okay? All right, today we're going to start talking about viral genomes. You know what genomes are, of course, the genetic material. We're going to talk about the different kinds of structures there are in viruses. And then we'll talk about genetics, how experimentally we can manipulate these genomes to do various things, to understand viruses, to prevent infections, and to prevent other diseases as well. Again, remember we talked last time in the 1950s, there was a breakthrough in virology where it was found that the nucleic acid genome carries the information needed to make new virus particles. So it's the genetic code. And that was previously figured out for DNA and bacteria in the 40s. But viruses were perceived as different, and it wasn't until the 50s that this was sorted out. So this is very obvious to us now, right? Nucleic acid is the genetic information. But back then it was a debate. It's very interesting. Things change. And the experiment we talked about the other day, the Hershey-Chase experiment with bacteriophage T4, which is this one right here, was an important part of that conclusion that the nucleic acid is the genome. But also work with tobacco mosaic virus, which we'll talk about in another lecture. And this is the first virus to be discovered. Remember, it's the virus that causes the mottling on tobacco leaves. So over the years, as people studied virology, they discovered thousands of different kinds of virions, different infections of all forms of life. And, but in the end, it turned out there are only a finite number of viral genomes. And that's going to be a focus of what we talk about today. There are just 7 different genome types. So commit that number to memory. It's very easy, lucky number. 7 genomes. So if I ever ask you, how many different viral genome types are there? Dr. Silverstein. 7. Not a lot. 7 different genome types. And that's it. And that makes it easy. And they're either DNA or RNA. That's it. As far as we know, nucleic acid doesn't come any other way. 7 types, DNA and RNA. This is why I teach virology. The composition and structures of these 7 genome types are more varied than anything else in any other kind of organism. Us, bacteria, fungi, protozoa, whatever. More varied in viruses than any other. That's why they're so interesting to study in part. And these are very simple organisms, if you will, but the decoding tactics are diverse. We see every possible tactic for encoding and encoding information. That is, the nucleic acid encodes information, it encodes protein. It also has non-coding regions that are important, which we'll talk about. But there are many ways you can do this, and viruses have done it all different ways. And in terms of molecular biology, the genomes have provided an almost unlimited source of subjects to study. Whenever we study a virus, we discover something new. So the amazing diversity is very fulfilling. The RNA and DNA genomes are structurally very different. Here are some of the different kinds of structures that we see in viral genomes. So they can be linear, which means a single molecule with 2 free ends, as opposed to a circle, where the ends are joined, of course. They can be segmented, that is, they can be in pieces. They can be 2 or 4 or 8 or 10 different pieces in the virus particle. They can be gapped. We will see examples of that. In other words, mostly double-stranded, but in some parts it's just single-stranded because there's a gap on the other strand. It can be single-stranded positive polarity. They can be negative polarity of single strand. It can be ambisense, which means they have both polarities on the single strand. We'll see how that works. They can be double-stranded as well. And some have proteins attached to the genomes, and some have the ends cross-linked. We'll see examples of all of these as we go through talking about specific viruses. And some even have DNA with RNA attached to them. So this does not occur in any other organism except viruses, this diversity of genome structures.
Segmented, do you mean they would have like separate
Pieces.
chromosomes, but like
Not chromosomes, because that's got a specific meaning, but they have different pieces of genome packaged in the particle. And they're all important for the virus to replicate. Not just one piece of DNA or RNA, but multiple pieces. So that's what we mean by segmented. Now, why is the question? Why are there so many ways of doing things? Mammals have double-stranded DNA genomes. Well, our mitochondria have some single-stranded circles, but for the most part, our coding information is in double-stranded DNA. But why do viruses have such diversity? Can it tell us something about the biology of the virus or how they have evolved by studying this diversity? Is one way of encoding information better than another? So is double-stranded DNA better than single-stranded RNA? Maybe for you, the most important thing is, do we have to memorize all of this? You don't actually have to, because after you understand, if you start at 7 different genome types, it will all come from that. And so you don't really have to memorize it, but I will ask you how many different genome types there are. Some of these questions are hard to answer. Why is a given structure in a genome used? Well, sometimes that reflects how the genome is packaged, how it's expressed, and we'll give some examples of that in the course. So a certain structure in a genome may be important for the replication cycle, and that's why it's present. But we often don't know why things are there. Why are there still double-stranded viruses and double-stranded DNA viruses and double-stranded RNA viruses? We have no idea why they're coexisting. Now, it's an interesting question why we have viruses with both kinds of genomes. As I said, we and other mammals have DNA-based genomes, but the RNA world is different. And there is a hypothesis called the RNA world hypothesis, which say that RNA genomes were the first to appear in evolution. So out of that primordial soup many, many years ago, the first genomes to appear were actually RNA. And there was a whole RNA world that lasted a long time where organisms were based solely on ribonucleic acid, not DNA. Today, the only RNA genomes left are viral genomes. No other organism has an RNA genome except viruses. So one question is, are these relics of the RNA world? So maybe viruses are really old, and they're giving us a glimpse at what was around in this so-called RNA world, which of course is hypothetical. And if so, if there was an RNA world, why was there a switch to DNA? And why did that predominate for all forms of life except viruses? We can't answer most of these questions, but they're very intriguing. And these are not the kinds of, I wouldn't ask you on an exam, why did DNA supplant RNA? Because we have no idea. All right, but it's an interesting. It would be interesting, but that would probably be for another course. So you need to memorize some of this stuff. You need to memorize the 7 different genome types, but as soon as you do that, how they work will be evident. So what information is in a genome? Well, gene products, proteins, and signals that we need for replicating the genome, obviously, for assembling the capsid and packaging the genome into it, and also things to regulate the timing of the replication cycle. You will see in the examples that we talk about, the replication cycles of many viruses are regulated. Things don't just happen all at once. For many viruses, they happen in a very orderly fashion, and that's encoded in the genome as well. There are gene products to modulate host defenses. As we mentioned briefly the first time, viruses wouldn't be around if they couldn't overcome host defenses. We have great host defenses, and there are a lot of proteins that have this job. And finally, the genome encodes proteins that allow the viruses to spread to other cells and hosts. So all these things are encoded in the genome. But there are also things that are not encoded in genomes, and looking at those can be instructive as well. For example, no virus has a complete protein synthesis machinery. The Mimi viruses, the really big viruses, have some genes encoding tRNAs and other parts of the machinery, but no one virus can translate on its own, and that's why they're parasites of the host translation system. Some viruses, as I said, encode RNAs and proteins involved in translation. There are no genes encoding proteins involved in energy production, which viruses require, of course, or membrane biosynthesis. So viruses require energy to replicate, and they can't make it. They can't make ATP. They have to get it from the host cell. Many viruses also use membranes. They can't make those either. They depend on the host cell to make those as well. There are no classical centromeres or telomeres that are found in standard host chromosomes. So the telomeres at the ends of the chromosomes in our genomes, for example, we don't find an exact duplicate of those in viruses. But in all these cases, you could argue that we haven't found them yet. So this is what we know, but it could be in a year or 10 years we find viruses that are exceptions to these rules. But as we stand today, we don't have genes encoding any of these things. Okay, so this will make your life easier, and I told you this before. Viral genomes must make mRNA that can be read by host ribosomes. If you keep that in mind, you will be able to trace the expression of a genome knowing what the genome structure is. And I'll show you how to do that over the next few lectures. So keep this in mind. There's 7 genome types, but they all have to lead to mRNA. And that's what Baltimore used to make his very interesting classification in the 1970s, where he took all the genome types and showed how they led to mRNA. So mRNA is central. You have to make mRNA because you have to make protein. As far as we know, the only way to make protein is through a translation of mRNA. So these are the 7 genome types, and as I said, he originally noted 6 because that's what was discovered at the time. And then later on, the 7th genome type, which is the gapped double-stranded DNA of the hepatitis B viruses, was discovered. So this is a very good scheme to understand and perhaps even commit to memory because it will help you understand how the viruses replicate. So some conventions also that you should memorize. mRNA is always the plus strand. By convention, again. Someone decided X years ago, we're going to call mRNA the plus strand. Someone asked me once if it has to do with electrical charge. No, it doesn't. Negative has nothing to do with charge. It's just mRNA translation plus. It sounded good. But in another universe, it could have been minus, right? DNA of the equivalent polarity is also the plus strand. So if the DNA, if it were RNA, and it could be translated, it would be the plus strand. So that's key, DNA of the equivalent polarity. RNA and DNA complements of that plus strand are simply called negative strands. We use this a lot in virology. That's why we're going over it again. Now, mRNA is ribosome ready. So mRNA is plus stranded. It can be translated directly into protein. But not all plus RNA is mRNA. Not all plus RNA has the structure that allows it to be translated into protein. But if it's an mRNA, it is automatically plus-stranded. So here is the secret to success in virology. There are only a few ways to copy these genomes, these 7 genomes to produce mRNA. The number of ways to do this aren't infinite, and that makes your life easier because you can remember them all. And in fact, it's logical. Knowing a few facts, you can deduce the pathway from any genome type to mRNA. The key here is that each pathway, whether we're starting from double-stranded DNA or single-stranded RNA, has its own polymerase, the nucleic acid enzyme that copies the nucleic acid. It has a primer, it has a template, and termination requirements. So each pathway, again, from those 7 different nucleic acids, have unique combinations of these things. And you can deduce those, really, once you know the rules. Each virus family sticks to one strategy. So a virus family is made, in part, the classification is done according to the nucleic acid. So the picornavirus family, for example, all have plus-stranded RNA genomes. So they will all follow the same strategy from their genome to making mRNA. So you don't have to worry that within a family you're going to have to understand different strategies. We will typically say, okay, an example of a picornavirus is poliovirus, and we're going to teach you how this virus replicates in a cell, and that will apply pretty much to all the other viruses in the family. Now, this is a particularly nice figure that I found which shows these 7 classes of genome, and it shows how they replicate. So here are 3 different kinds of viruses. Viruses with RNA genomes, viruses that reverse transcribe their RNA. That means they take the RNA and they make a DNA copy. And then there are the viruses that have DNA genomes. And the RNA viruses here, genome replication cycle, this is a nice cartoon that shows you that all you have to do here is go from RNA to RNA. So the virus RNA comes in the cell, has to make more RNA and more RNA, et cetera. Quite simple. The contents of the virions, the RNA viruses, can be plus or minus single-stranded RNA or double-stranded RNA. So that breaks it down, makes it very simple. And then at the bottom, there are some examples of the kinds of viruses that fall in here. All right. Then we have our reverse transcribing viruses. These are viruses with an RNA genome or, in some cases, a DNA genome. And they go back and forth from one to the other. But again, within a virus family, it only goes in one direction. So the retroviruses have an RNA genome. They make DNA out of it. The hepadnaviruses have a DNA genome. They make RNA and then DNA using reverse transcriptase. Okay, so the reverse transcribing viruses simply means you go back and forth between RNA and DNA. And these are 2 examples, retroviruses like HIV and the hepatitis B virus. They have different genomes in the virion. The retroviruses have an RNA genome that's plus-stranded. Now you may say, why aren't these the same as these plus-stranded RNAs? Well, because these have a reverse transcriptase and they have a DNA phase in their life cycle. These viruses do not. And finally, we have DNA viruses that simply go from DNA to replicate their genomes. And these can be, in the virion, either single-stranded DNA or double-stranded DNA. They can be circular or linear. So I find this useful to summarize this with some examples that goes beyond the Baltimore table. So I think you ought to memorize the 7 genome types, but you can already do it almost, because if you just think of every possible configuration of nucleic acid, you already have it done. And that will help you understand how the genomes are expressed. So don't wait till the night before the first exam. You should start now. You should start thinking, what are the 7 types? Can I remember them? Because if you know the structure, you can deduce how mRNA is made and how the genome is copied to make more genomes. And that's something that I want you to know at the end of this course. Now, the textbook is useful for this because it has a lot of illustrative material that helps. It has, so in Chapter 3, the chapter is called Genomes and Genetics, which is what we're talking about today. It has figures which summarizes everything I've told you. And in that chapter, for each of the 7 genome classes, there's a box that has essential information. It tells you the genome configuration, how much the virus depends on the host for replication. This is a key point that we'll be talking about a lot. Some viruses are very dependent and others less so. And gene expression strategy. So all of this is in Chapter 3 following each of the 7 genome strategies. And this is an example of one of those figures. So at the top of this figure, you have single-stranded DNA genomes. So that's one of the 7 Baltimore classes. It has a little picture of the single-stranded DNA, 2 examples of families of viruses that would have this kind of genome. And you can tell that a word is a family name because it has a E at the end, Cercoviridae and Parvoviridae. And then at the top here, it has the strategy by which the genome is not only made into mRNA, but is also made into new viruses. So here, these single-stranded DNA genomes, they can be actually either minus or plus stranded. They're copied to double-stranded DNA. Now, you may ask, why would you automatically assume that you have to go from a single to a double-stranded DNA genome? Well, you have to know one fact, and that is you cannot make mRNA from single-stranded DNA. So if you know that, you'll be able to figure out when you see a single-stranded DNA genome that the first step that has to happen is it has to be converted to double-strand. So that's what happens. You make double-stranded DNA, which can be then transcribed to our mRNA. It's there. That can be made into proteins. Then the genome has to replicate. We know that we came in with single-stranded DNA, so we have to make more of that to put into new virions. So the genome is replicated to plus and minus strands, and then it's packaged into capsids. So that's all embodied in this one figure at the top. And then on the bottom are 2 examples of the genomes that we've shown up here. The Cercoviridae are single-stranded circles. It tells you the size, but I don't expect you to memorize genome sizes. I just want to give you a sense for how much it varies. So circular single-stranded DNA, degree of dependence on the host, how the gene expression works, and some other features which might be of interest. And unless I point these out in lecture, you won't need to know those. And here's another kind of virus with a linear single-stranded genome with interesting structures at the end. And we'll talk about how those work. So this is in the chapter for all 7 genome types. So I would have a look at that, and I think if you just start with the 7 genome types memorized, you'll be able to deduce everything else from it. So here are the 7 classes. I just want to go through them one by one and talk about how they work. Double-stranded DNA, gapped double-stranded. So you can put them into 2 columns. You have our DNA and RNA and single-stranded DNA genomes. And then we have RNA genomes, double-stranded RNA, single strands of plus or minus polarity. And then our plus RNA viruses that go through DNA intermediate, the retroviruses. Okay. That's where the name comes from, retro, because they go from RNA to DNA, whereas for many years we always thought the flow of genetic information was DNA to RNA to protein. But this was the wrong way. Well, it's not the wrong way. It was discovered. My expectation when you are done with this course, you will know the general answers to these questions for each genome type. So this is what I want you to know. What are the steps to mRNA production from each genome? If I say, I give you a single-stranded DNA genome, how do you get to mRNA? It's easy. You tell me, well, you make a double-stranded DNA copy, and then you transcribe that to make mRNA. And depending on the virus, we will either use viral enzymes or host cell enzymes. So those are the steps. What general gene products must function at each step? So to make single-stranded DNA into double-stranded DNA, the host will do that for most of these small viruses. And then to make mRNA from DNA, depends on the virus. Some viruses will use host enzymes, and some viruses will use your own. When in the life cycle is mRNA made? So DNA viruses, when they first come in, there's no mRNA present, so they have to make it. And they can either make it early or later. Other viruses come in, RNA viruses, they're already mRNA. So it doesn't have to be made initially. How's the genome copied? What kind of enzymes are used? What kind of replication proteins? Again, are they viral proteins or host proteins? And which proteins produce mRNA? And how is this information converted to gene products? For the most part, we have a single mRNA. It's translated into protein. But as we'll see in the translation lecture, there are many variations on that viruses have devised. I would also suggest you look at Appendix A. This is full of figures like this. This is a replication cycle of a hepadnavirus. And it shows you all the steps. And you get a very nice overview of what has to occur. So you can see the viral genome coming in and it's getting into the nucleus. And you'll find out in the lecture that all the gaps in this genome are repaired because do you think you could make mRNA from a gapped DNA, double-stranded DNA? Just guess. No, you can't, because you can't make it from single-stranded DNA templates, and you can't make it from gaps. So you have to repair it, and then this can be transcribed and translated into proteins. So you'll see here viral life cycles, some facts about the genome, and you can relate the simple strategies to the life cycles in this way. You can say, okay, I start with a single-stranded or double-stranded or gapped genome. What do I have to do? It's all summarized right here. I think that helps a lot. So let's look at some of these genomes one by one. The first are the DNA genomes. As you know, our genome is based on DNA. We've already said that. So many DNA viruses, for the most part, emulate what we do with our DNA. But viral DNA genomes are mostly not like cell chromosomes. Some of them have features of cell chromosomes, but they're not like our chromosomes. So I would prefer if you did not call them chromosomes, but I understand that it may be ingrained in your lexicon, but they're not chromosomes. Unexpected tricks have evolved, which we don't find in cellular genes, and we'll point those out. So some questions to get you thinking about DNA genomes. How many ways can you think to replicate DNA? Do DNA viruses always replicate in the nucleus? Well, you know, if a virus is going to use host DNA replication enzymes, it has to go into the nucleus, because that's where they are. But some viruses don't have to do that. And we'll tell you how to figure out which ones do it and which ones don't. Most cells in your body are terminally differentiated, in fact, do not replicate their DNA. So what happens to a DNA virus that needs the enzymes of the cell when they try and infect one of these cells? We'll tell you what happens there. But these are interesting questions to think about already. Do DNA genomes always encode their own DNA polymerase? Are there viruses with DNA genomes that encode no DNA polymerase? And we'll answer all of these in the coming weeks, but I think these are quite interesting. And finally, do DNA genomes exist that do not require a DNA polymerase to copy them? So let's start with double-stranded DNA genomes. There are 22 families of viruses. I'd never ask you how many families of double-stranded DNA viruses there are, but I just want to give you a sense of how many families. This is probably an underestimate because we're discovering viruses by the hundreds now, and there are probably many more families. Here are some of these viruses. Adenoviridae, these are all family names. Hepadnaviral, herpes, papilloma, polyoma, pox. We'll talk about some of these and how they replicate and cause disease. The topology of the genome is critical for the life cycle. So double-stranded DNA genomes can be linear or circular. And these topologies, as you'll see, have major implications for replication. I've already told you one, if they're single-stranded, you have to become double-stranded before you can make mRNA. Making mRNA, information extraction. mRNA is produced when either a host or a viral enzyme copies the minus strand. Now remember, in a double-stranded DNA, the plus strand is the strand that's the same polarity as mRNA. So that means to make mRNA, you have to copy the minus strand, right? So remember that. It can be confusing. You cannot make mRNA from single-stranded DNA, which I have already told you. And you can only make it from double-stranded DNA. So single-stranded DNA or gapped DNA, you cannot make mRNA from. So you can immediately deduce that you have to sort out how to get to a double-stranded DNA template for that. So here are some double-stranded DNA genomes. Here in the virion, they're double-stranded DNA molecules. They can be either circular or linear. Here's an example of a family, the polyoma viruses and the papilloma viruses, which have circular double-stranded DNA genomes. The adenoviruses, herpes, or pox viruses have linear double-stranded DNA genomes. These can be transcribed directly into mRNA, so that's the first thing that can happen in a cell. The DNA is copied to mRNA. Proteins are made. And depending on the virus, some of those proteins participate in DNA replication. Some of the biggest viruses can encode their own DNA replication systems, but many need help from the host. And some proteins have to be made. So then it's very simple. You make mRNA from the DNA. You make proteins that participate in replication, regulation of the cycle, and eventually formation of virions. So those are double-stranded DNAs. We have double-stranded DNAs which are gapped. The gapped DNA genomes, and these are the genomes of the hepadnaviruses. The human pathogen here is hepatitis B virus, which causes liver infection. serious human pathogen. These have a very interesting genome, which is schematized here. First of all, there's a protein covalently linked to the 5' end of one strand. So in the book and in all my slides, I have color conventions. So DNA is always blue. One strand is lighter color than the other. RNA is green, 2 different colors for plus and minus. And then proteins are some kind of earth tone, I think. But the nucleic acid's important. So you can see here, this is double-stranded DNA. You can see there's a gap here. You can also see this protein linked to the 5' end of one of the strands. And also there is a piece of RNA linked to one of the strands as well. Short piece of RNA, which you can tell it's green. So that's the gap DNA with its short RNA. So one strand is complete. This light blue strand, the minus strand, is complete. And the other is only about half completed. So that's why we call it a gapped DNA. So that can't make mRNA, as we've already said. When it comes into the cell, it has to be repaired. And as far as we know, that uses cellular enzymes. So the hepadnavirus DNA genome is repaired, converted to double-stranded DNA. So repair means you take off the protein, you take off the RNA, you fill in the gap, because you need perfectly duplex DNA, double-stranded DNA, in order to make mRNA. So you have to remove those RNA and protein, you have to fill in the gap and close the ends, make sure they're ligated together. And all that has to happen before mRNA. And this DNA genome, we will see in a subsequent lecture how it's produced during replication, why it ends up with proteins and RNAs and gapped. But the enzyme that is involved in the replication of this virus is also a reverse transcriptase, very similar to the enzymes that are used by retroviruses to copy their RNA genomes into DNA. All right, so here's our double-stranded gapped DNA genome. Just to give you the whole scheme, you start with this gapped genome with the RNA and the protein linked. You repair it, and then it can be used to make mRNA, which is used to make proteins. Now, a curious thing happens during the life cycle, which you couldn't predict. So this is something you'll just have to know. There is another kind of RNA made by transcription, which serves not as a template for protein synthesis, but for making first a negative strand of DNA and then a double strand of DNA. And that's eventually what will get incorporated into the virion. So that's the reverse transcriptase that takes this RNA and makes it into DNA. Now, this is really Baroque, right? You would say, why does this virus do this? Because there are other ways to get replication done, right? We don't know the answer. It works. That's what I always fall back on. Whatever works for viruses in an evolutionary sense is there. So this has survived eons of selection, and that's all we can say about it. Single-stranded DNA genomes, there are 5 viral families in one genus, and there are viruses that maybe you've heard of, the parvoviruses. If you have a dog or a cat, you have to get them vaccinated to prevent parvovirus infection. Can kill them. Circos you may not know much about, but probably all of you are infected with one of these viruses, and you're fine because they don't seem to cause any disease. They have single-stranded DNA genomes, and of course, as we said, they cannot be made into RNA. So we have to make them double-stranded before we can make mRNA. Now, these are very small genomes. So the only reason you might want to learn that these genomes are small is because if they're small, they're very unlikely to encode a DNA polymerase. So these genomes usually encode a protein that is used to build a capsid and one other protein that's somehow involved in replication but is not a DNA polymerase. So these small circovirus, parvovirus genomes don't encode DNA polymerases. So that means they need to use the host DNA polymerase. So now, what can you tell me about where the viral DNA goes when it first infects a cell? So it's single-stranded. Where do you think it has to go? So it can't make mRNA until it's made double-stranded. So where do you think it has to go? To the nucleus, because that's where the enzymes are that replicate. So that's what I mean by applying a few facts. You can logically think through these processes. So all replication in these viruses is accomplished by cellular DNA polymerases, which are in the nucleus. So that's where these viruses have to go. So here we are, single-stranded DNA genomes in our Chapter 3 scheme. Single-stranded DNA is converted to double-stranded DNA by a cellular polymerase. It happens in the nucleus. You make mRNA in the nucleus. Now, you wouldn't be able to guess this, but if a virus doesn't encode of this size doesn't encode a DNA polymerase, it's not likely to encode an enzyme to make mRNA as well, and these don't. So these transcriptases, the enzymes that copy DNA into mRNA, are the ones used from the cell, and they're present in the nucleus. So we make viral proteins to do more replication and make more infectious virions. The circoviruses have a single-stranded circular DNA genome. It's very small. It's among the smallest DNA genomes that we know about. Yeah?
I'm just wondering, so what happens to the matrix strand DNA at the end of the
Well, some of these viruses actually package both strands. So in some cases you use them. If it's a double-stranded DNA, of course, both strand goes into the virion. But I think a good fraction of these are actually used, and those are not. They're degraded. Yeah. Yes?
What did you say was the other thing that the genome would encode for besides DNA polymerase because they're so small?
So the smaller viruses don't encode a DNA polymerase or a DNA-dependent RNA polymerase, the enzyme that makes mRNA. I have to remember to repeat the questions so you can hear them on the tape. Okay. So this is single-stranded DNA, very small. These are slightly larger, the parvoviruses. And as I said, the circos are ubiquitous. You're all seropositive for these viruses, but we don't know that they cause any disease. The parvoviruses do cause human disease called fifth disease, and it's a rash-like disease, a rash disease of humans. And they also cause disease of cats and dogs. They have a very unusual genome structure, which we will talk about when we talk about DNA replication. How about viruses with RNA genomes? Now, cells, these are unusual because cells have no enzymes that can copy these genomes. So it's a little easier for you to remember how they work. Just remember that RNA cannot really be copied by cells. And I say really, you may think, well, what is he qualifying there? The cells can copy short pieces of RNA, right? They can make microRNAs, and the whole small RNA system in cells is made by copying small pieces of RNA. But they can't copy RNA genomes. Whatever enzymes do that cannot copy big viral genomes. So that's why we say cells have no polymerase to replicate these genomes and to make mRNA. So they have to all use their own enzymes. These viruses, no matter how small they are, if they have an RNA genome, they have to encode an enzyme to copy it. So they encode novel polymerases. Novel meaning the cell doesn't have anything like it. So if you make an inhibitor of that polymerase, it's a good antiviral drug, or in principle it's a good antiviral drug because the cell doesn't have anything like it. Whereas DNA polymerases of viruses are quite similar to cellular polymerases, and there can be some effect of antiviral drugs that target viral DNA polymerases on the host cell. So these RNA polymerases encoded by these viruses, they replicate the RNA genome and they make mRNA. And we will talk about this in detail. So just to tell you that you don't have to understand it at that point. They're two different processes. The mRNA, of course, has to be read by ribosomes. Otherwise, you can't make proteins. So we have all sorts of configurations of RNA, too. We have double-stranded RNA. And there are 7 virus families with viruses of double-stranded RNA genomes. Many of them are segmented. We come back to this word segmented. I'll show you a picture of a segmented genome, but not all of them are. Some of them are unimolecular, one RNA in the virion. Here's an example of a virus family with segmented RNA. These are the reoviruses. They have 10 to 12 separate segments of double-stranded RNA. And these include rotaviruses, which are huge causes of gastroenteritis. There's also another family that infects invertebrates with 2 segments. So the Rios have between 10 and 12. And I don't mean by saying 10 to 12 that within a given virus, some of them have 10 and some of them have 12. It's just within a certain species of virus within this family. Some will always have 10 and others will always have 12. Here's another factoid, which is good. So single-stranded DNA cannot be transcribed into mRNA. Double-stranded RNA cannot be translated by ribosomes. Even though there is a plus strand in that double-stranded RNA, cannot be accessed by ribosomes. They don't know what to do with this double strand, so there's no enzyme to pull that apart. So the viruses have to have a way to make a plus strand, a single-stranded plus strand from this duplex RNA. So here's an example of a reovirus with 11 double-stranded RNA segments. So this is a segmented genome. They are here in this case 3, 6, 9. There are 2 more not shown here. 11 segments. And they're all packaged into the virus particle. So you need all of them for infectivity. And again, because the double-stranded RNA can't be translated, the first thing that has to happen is you make mRNA, which can be made into protein, and that can be used to make more RNA and more virions. So you see again what I mean by a few facts leading you to figure out how the replication works. Single-stranded RNA viruses with a plus strand RNA genome. There are a lot of families like this, 22 that we know of. 8 of them have viruses that infect mammals and are significant pathogens. So the picornavirus, poliovirus, and rhinovirus are significant human pathogens. The caliciviruses causing gastroenteritis. Astrovirus also gastroenteritis. SARS coronavirus. And then there are flaviviruses of medical importance, yellow fever, West Nile hepatitis C virus. Included in this classification are retroviruses, but they're actually a separate category in the Baltimore system. That includes HIV and HTLV, and then rubella and other encephalitis viruses are also in this categorization. So here's how these viruses work. The genome is plus strand, so it can be translated. It can be translated into protein. So the genome comes in the cell, protein is immediately made, and that protein is eventually used to make more virions. The genome has to replicate, and to do that it has to go through a negative strand intermediate. So that's the way nucleic acids work, as you know. To be replicated, you make a copy of one of the strands, and the same goes on with RNA viruses. You take the plus strand genome, you make a full-length minus strand copy, which has no function other than being a template for the synthesis of more plus strands. So eventually those minus strands are degraded, they're not used because they're not packaged any further. What goes into the virions are the plus strands. So these are some examples of these genomes. Some of them are very long. The coronaviruses, where SARS is a member, 28 to 33 kilobases. These are the longest RNA virus genomes that we know of. And this is an interesting question. Why is there this limit on RNA, the size of RNA genomes? DNA viral genomes can be 1.2 million bases long. But this is the biggest for RNA is 33 kilobases. We're not quite sure why. They have unusual features at the ends of the genome, which we'll talk about here. For example, the picornaviruses have a protein link to the end. Many, but not all of these are also polyadenylated. So these are messenger RNAs. They roughly look like mRNAs in the cell. They have a cap and a poly A. But again, not all of them do. The flaviviruses don't have a poly A, the 3' end. The picornaviruses don't have a cap at the 5' end. So this is what I mean by viruses doing unusual things. And we'll talk about how those things work later on. So one of these viruses that we had on the previous slides were single-stranded plus RNAs that go through a DNA intermediate. So there's one viral family that fits that description, the retroviridae, and there are 2 pathogens in there, HIV and HTLV. Maybe there are more. There's another being considered, but we're not sure yet. And the strategy is remarkable only because when it was discovered, which was in the 70s, nothing like it had ever been seen before. So the plus-stranded RNA is in the virion. Now, here is a little exception to your logic. You would say, well, that has to be translated into protein because it's plus-stranded RNA. Well, in fact, for these viruses, and that's something you'll just have to remember, that plus-stranded genome does not get translated. It is not translated in the cell. And it gets copied into a minus strand DNA copy by an enzyme in the virus particle, the reverse transcriptase. It's then made double-stranded. And now that you have a double-stranded DNA, now you can make mRNA, right? And you can make mRNA that can be made into protein, and you can also make RNA that goes into the genome. So the RNA in the virion is a real mRNA. It's plus-stranded, looks just like an mRNA. If you extracted it from the genome, you could translate it into protein. But it's never used as a message in infected cells. It's converted to DNA by this enzyme reverse transcriptase. And what's more interesting here, this double-stranded DNA copy is not floating around in the cell and then copied into mRNA, as is the DNA genome of many other viruses. But this DNA integrates into the genome of the host cell. And there, the host RNA polymerase copies it to make mRNA. We will go through that in a separate lecture entirely, but it's very different from the way all the other viruses replicate. So the proviral DNA, and proviral means the DNA copy of the virus that's integrated into the host cell. That serves as the template for mRNA synthesis and, in fact, viral genome synthesis. So here, this DNA copy is integrated in your chromosomal DNA. In fact, all of you have these in your genomes. Fortunately, they are not making retroviruses. They're mutated extensively. But you all have these, and in other animals where they're not extensively mutated, they make mRNA, and they make another copy of RNA that gets incorporated into virions, and that's done by the cellular polymerase. So then we move to some minus strand RNA viruses. There are 7 virus families with negative sense RNA, and these are some of the mammalian viruses. Measles and mumps you may recognize, rabies, Ebola, Marburg influenza, and Lassa viruses. Some of these very serious human pathogens. Now, the minus strand RNA, you can predict that when it gets into a host cell, you know it can't be translated by definition because it's minus strand. So what has to happen? It has to be copied into a plus strand. And in addition, because you know that cells cannot copy RNA, that copying has to be done by a viral enzyme. So knowing a few things, you can predict what is going to happen. So these negative stranded RNAs are copied to mRNA by the virion enzyme, which is then translated into protein. And of course, you need to make more negative strand RNAs to put into virions, and those are done through a replicative cycle, through a plus-stranded intermediate. And these enzymes, by the way, that copy this, have to be in the virion itself. Because if this negative strand came into a cell without the enzyme, nothing would happen. Even if the enzyme were encoded in the genome, the cell cannot access the coding potential in that genome because it's a minus strand. So the enzymes have to be in the particle. So I could say to you, you have a negative strand virus with a single-stranded RNA genome, negative polarity. What is in the particle? And you can tell me there's an enzyme to copy that. You will absolutely know that by automatically knowing these facts. So there are no enzymes in the cell that can make RNA, so we have to use a viral polymerase to do that. And that polymerase makes mRNAs, and it also replicates the genome, which means basically it makes a full-length plus strand that is not a message. It's reserved for replication. And those are templates for making the minus strand RNA genomes. So here are some configurations of genomes of single-strand RNA minus sense viruses. Some of them are segmented, just like those reovirus genomes were question? Yeah?
Yeah, do you have a single-stranded minus strand virus that's retrovirus that'll make DNA first and then?
So the retroviruses have single-stranded RNA genomes of plus polarity.
They always have to be plus.
Well, that's all we know of. Yeah, we haven't discovered any minus polarity. But if you had a reverse transcriptase in the particle, it's theoretically possible, right? Because the reverse transcriptase could copy the negative strand into DNA. So there's no theoretical reason why that couldn't occur. But we've never found that. Right? So we have segmented genomes. So here, for example, influenza virus genomes is in 6 to 8 segments. So again, influenza A viruses have 8 segments of RNA. You need all of them in the virus to be infectious. They're all single-stranded. And then we have some viruses with non-segmented genomes, paramyxoviruses, mumps, measles virus, rabies virus are all single-stranded RNA genomes. So 2 different configurations. Now, having a segmented genome allows you to do something that viruses that don't have a segmented genome can do. What am I saying? It allows you to do things that genomes that are not segmented cannot do, right? And that is something called reassortment. So when these are 2 different influenza viruses infecting a cell, and when these 2 viruses infect the cell, each of them has 8 RNA segments, and they can all mix within the cell, and they replicate, and so you have a mixture of 16 different RNA segments from 2 different viruses. So the viruses that are produced can be either of the 2 parental viruses or viruses that have mixed segments. They all have to have 8, and they all have to have 8 specific segments, but they can come from either parent virus. So this is a way that these viruses undergo genetic mixing, if you will. It's called reassortment. All right, so it's a very specific term. It applies to segmented viruses. Now, when nucleic acids recombine, you probably have learned this in other courses. You can have DNA recombination, breakage and reunion of DNA, or copy choice mechanisms that allow the shuffling of pieces or sequences of DNA among different organisms. But viruses have another way to do it with having a segmented genome. These can readily mix, and the frequency is higher because you don't need breakage, you don't need copy choice in order to do this. So this happens with influenza, and it's probably a major reason why these viruses have been with us for many years, and it's very hard to get rid of them. Some of the minus strand RNA viruses are called Ambisense. That is, they have both plus and minus aspects. And there are 2 families of viruses, Arenaviridae and Bunyaviridae. And the Lassa viruses are in the Arenaviridae family. These are very serious viruses that cause hemorrhagic fever, highly fatal, but largely restricted to a few countries of the world. So here's what these genomes look like, Ambisense RNA genomes. So you can see, I think, the 2 colors of RNA. We have our light green color, so this is Kelly green, and this is olive green. Okay? And that's because they're 2 different polarities. We have the plus sense here, and then the rest of the genome is minus sense. So it's actually a mixture of polarities. So this part of the genome could be translated, but not this part because it's negative polarity. So this virus that we're illustrating here has a genome of 2 RNA segments, an L segment and an S segment. They're both ambisense, and they encode 2 proteins for each segment. And what happens is when these viruses infect the cell, the first thing that in fact happens is that there's a viral enzyme in the particle that makes a small mRNA complementary to this negative sequence at the 3' end. So again, the olive green part is minus strand, so you can't translate it. So a virus enzyme makes an mRNA, which is now the plus strand, and that can be translated into protein. On the other hand, the sequences at the 5' end can be directly accessed, but they're not. In fact, a copy of this whole genome has to be made. So now this goes from plus to minus, and then this is copied into an mRNA from which proteins can be made. And probably the reason this isn't translated is because there's no cap here at the 5' end. So that's an example of a segment that has both kinds of polarities in it. And I don't know why these have arisen. Again, they exist. Evolutionarily, they have survived, so it's a strategy that works. So here's a nice figure that summarizes everything. There's a very good site over in Switzerland. Here's the URL, which basically classifies viruses and has lots of very nice graphics, such as this one. So it has our 7 groups of DNA genome, of a viral genome, all the DNA and RNA types with nice illustrations. And they have a little cartoon above. And in fact, these are links that you can click on and go to descriptions of each of the different kinds. And you can see the flow of information from the genome through the intermediates to mRNA and eventually to protein. So this is another way of summarizing what we've been talking about. It's a modern view of the Baltimore scheme. Now, in the virus particle, what do the genomes look like? I've been drawing these linear diagrams, but those are mainly illustrative. Of course, we don't think this is what the RNA looks like in a particle. We don't really know what the RNA or DNA genome looks like because we can't see its structure. It seems to be largely disordered inside, and when it is disordered, you don't know what the structure is. But there have been some predictions which suggest that the genomes of some RNA viruses are all folded up and look like something like this. So the RNA I showed you first, this RNA, this is a picornavirus RNA about 7.5 kilobases long. In reality, this may be in the virus particle folded up in this way. And here is the very first base of the RNA here where this arrow is. So this is called RNA secondary structure. We'll talk about this more later on. But it's likely that in the particle, genomes aren't linear. They're folded in a variety of ways that are important for function. And in the text, there is a table I think this is in chapter 3, which shows some of the different configurations genomes take on. So we've already seen these very interesting parvoviral genomes, the gapped DNAs of hepadnaviruses, coronaviruses genomes, which are highly structured here and here, and a variety of others showing interesting structures. These all have functions in replication, some of which we'll talk about in detail. But it just serves to illustrate that in the particle, the genome is in a very specific configuration. It's not just a line, a squiggly line that I've drawn on the screen. Now, I want to talk a little bit about how we use genetic methods to study viruses and what we can do with that, and a few terms that we need to set forth because we'll use these a lot. Wild type we use often. This means the original virus from which you select mutants. It's often a laboratory adapted virus. We call it wild type because it's our starting material. But it's really an arbitrary name because we know now that you can take clinical isolates from different individuals and they'll all be slightly different. So it's an old term which probably isn't accurate anymore. This may or may not be identical to a virus you isolate from nature, but we use it as a starting point. Now, when you get virus isolates from a natural host, we call them field or clinical isolates. So often I'll be talking about a clinical isolate, and that's what I mean, something directly taken from an individual, as opposed to a virus that's used in the laboratory for many years and has been passaged extensively. Some of the methods we use in virology, in addition to the ones we talked about the other day, these pertain to the genomes. One important one is called DNA-mediated transformation. And this is the introduction of foreign DNA into cells. And the reason why it has a long name, DNA-mediated transformation, is because before this, it was appreciated that you could put DNA into cells and change their phenotypes, hence the word transformation. The word had already been used to show, to describe oncogenic transformation, the transformation of cells into an oncogenic phenotype. So hence, not to confuse the two, we put DNA-mediated transformation in front. Now, this is the abstract of the 1944 paper from Avery, McLeod, and McCarthy at Rockefeller, which showed that you could transform the morphology of a bacterium, in this case a pneumococcus, using DNA. So they took two different colony morphologies. Here, the two different morphologies, a small one and a large shiny one, and they could transform one to the other by taking the DNA of one strain and putting it into the other. And this was the first proof that DNA was the genetic material. So you see they called it transformation of pneumococcal types, and that's where the word DNA-mediated transformation came from. Now, nowadays we use another term, transfection. Transfection specifically refers to the production of virus after you put viral DNA into cells. So you take viral DNA, purified viral DNA, you introduce it into cells. That's DNA-mediated transformation. And that initiates an infectious cycle. So that's called transfection because it's transformation and infection, transfection. But unfortunately, people don't really like to say DNA-mediated transformation all the time. So they say transfection for everything. So you take a gene encoding a protein of any kind and you express it in a cell by putting DNA in the cell. They call it transfection. It's not really correct, but you will hear everyone, including Nobel laureates, using it. So I just want to explain what is the correct usage of the term. Transfection really refers to initiating a viral infection with nucleic acid. Mutation, of course, is a change in DNA or in RNA. Base changes, insertions, deletions, and rearrangements. We'll talk a lot about mutation in this course. Nonsense mutation, missense mutations, but it is not a change in protein. If you say, I mutated the protein, that's not right, because mutation means you're working on nucleic acid. And this is, again, something that people don't use correctly, so I want to tell you the proper usage. Mutation is for nucleic acid. What method allowed the application of genetic methods to animal viruses? Anyone wish to hazard a guess? Yeah, if you printed out the lecture notes, the answer is right there. What is that? What? Cell culture helped, but there's another more specific assay. You see that circle? What is that picture? Plaque assay. it allows you to pick genetically pure cultures of a virus, right? You need to be able to do that to do genetics. If you can't purify a virus, clone it, you can't do genetics. So yes, the plaque assay is crucial for this. So that's why it was such a big deal when Renato de Becco devised the plaque assay for animal viruses in 1952. It allowed you to isolate mutants of various sorts. And how did we do that back then? Well, it turns out that for RNA viruses, if you grow up an RNA virus stock, you have a tube of an RNA virus, it's full of mutants already because the polymerase makes mistakes. It makes about one mistake in 10,000 or 100,000 bases that it polymerizes. So many RNA virus genomes are 10,000 bases in length. So that means that every time it's replicated, one mistake is made. So a stock of virus, which has billions of viruses in it, is a wonderful collection of mutants. DNA viruses don't make as many mistakes, several orders of magnitude higher. So there are fewer mutants, but they're still there. You just have to work harder. And in some cases, you mutagenize the viruses with chemicals that introduce mutations so that you can get mutants where you can't otherwise. So some of the mutagens that were used are listed here: nitrous acid, hydroxylamine, and alkylating agents. They modify the DNA such that when a replication occurs, you get mispairing. So if you find any of these chemicals anywhere, don't ingest them. They're very mutagenic. They will cause mutations in your DNA. There are also base analogs that cause changes in DNA during replication. UV also does that as well. So all of these were used to mutagenize viruses to induce mutations when you couldn't find them naturally in a stock of virus. Now, let's say you take a tube of virus and I give you a tube of RNA virus and I say, get some mutants out of here for me. What would you do? Well, you can screen for phenotypes. You would ask me, well, what do you want? What kind of mutant do you want? Well, you could say, give me a temperature-sensitive mutant. That is, give me a virus that doesn't grow at a high temperature, like 39 degrees. We usually grow viruses at 37 centigrade. I say, give me a virus that doesn't grow at 39. And so that's easy to screen for in your collection of mutants. You could say, how about a plaque size mutant? How about a tiny plaque or a bigger plaque or some morphological alteration in the plaque size? Cold sensitive mutants, viruses that don't grow at low temperatures. You can readily devise screens to select out of that tube of RNA viruses or your DNA virus treated with a chemical of some sort, these kinds of mutants. Drug resistance, escape to antibodies. There are lots of examples of phenotypes that you can search for. But nowadays, we don't really do this except maybe in drug discovery because we actually engineer mutation into the genome. So we say we have a viral genome. We're going to put a mutation exactly where we want it and see what the phenotype is. And this is because we now have what are called infectious DNA clones for just about every virus that we work with. We have DNA copies of their genomes, which we can modify very specifically and get exactly what we want. And this is a modern validation of the Hershey-Chase experiment, which you may remember from the other day. Remember, Hershey-Chase said that the genome contained the information to make new particles. So if you take a DNA copy of the genome and you put it in the cell and you get virus out, that validates Hershey-Chase in a different way. And you can make all kinds of mutations in these DNA copies. Use recombinant DNA methods. The sky is the limit. You can do whatever you want. There's Hershey-Chase, which we talked about the other day. So we have infectious DNA clones for basically all viruses. DNA viruses, which are all shown here. RNA viruses, plus and minus strand, double-stranded RNA. Let me show you two examples of how you do this. Here's poliovirus. Polio has a plus-stranded RNA genome. When the RNA gets into a cell, it can be translated. So if you extract the RNA from poliovirus and transfect it into cultured cells, you will initiate an infectious cycle. So the RNA is infectious. You can take this RNA and make a double-stranded DNA copy of it and clone it into a plasmid, and then use the plasmid to make RNA in vitro with various enzymes. And then you can take that RNA and put it into cells, and you will get virus again, because this is very much like the viral RNA. This is great because you can modify this DNA. Again, you can mutate it. You can add bases, subtract them, change them. You could add sequences from other viruses. You can do whatever you want. You can't do that with RNA. So we can do genetics having DNA copies of viral genomes. You can do the same thing with influenza virus. Influenza virus has 8 negative stranded RNA segments. So what you have to do for that virus is you have to clone each of the 8 RNA segments into a plasmid, and then you engineer the plasmid, which is shown in a linear way here, to make both plus and minus strand copies of the RNA. The reason you have to do that we don't need to go into. But basically you take 8 plasmids, each plasmid encoding one of the viral RNAs. You put those into cells, 8 plasmid transfection, and you get virus out. So again, with influenza, you can do the same thing as with the coronaviruses. You can make changes in the virus and see what their effect is. Now, this technique was used to rescue the flu virus of 1918. We didn't have cell culture in 1918. You know, there was a huge pandemic back then. Millions of people died, but we don't have the virus from it. It's a shame. We would like to study it. So what people did was they resurrected it, because we didn't have flu until 1933. In 2005, viral RNA was isolated from formalin-fixed lung samples from someone who had died in 1918. So the Army, you know, a lot of military people died of flu in 1918-19. And whenever they died, the Army would take sections of their lungs and they would put them away. They're very good at keeping these sections. They're all in order. So someone went back to those archives, took some of those sections on slides and took off the tissue and extracted RNA, got the sequence of the RNA. They also got RNA from someone who had died of flu in 1918, and they were buried up in Alaska. The ground is frozen all the time. So they opened the grave, and they did a biopsy of the lung, and they took the tissue, extracted the RNA, and sequenced it. So they could reconstruct the sequence of the entire virus. So they made 8 plasmids of the 1918 virus, put those into cells, and they got the 1918 virus out. Complete sequence, and the virus is recovered. Of course, you have to study this under very high containment. You can't just do it up the street here. You have to do it in a BSL-3 facility because it's a very lethal virus. But this shows you how you can reconstruct viruses from viruses that we don't even have anymore. Now, the other thing you can do with having cloned viral genomes and the ability to resurrect viruses from DNA is you can make vectors. And vectors are good for research, but they can also be used to treat human diseases. So in gene therapy, the goal is to deliver a gene to patients who lack it or have a mutation, and you can use viral vectors to do that. So one way to do it is to take cells from a patient, infect them with a virus vector that expresses the gene, and then put them back into the patient. You can use these vectors to deliver antigens. In other words, you can make a viral vaccine from such vectors, and we use them in the research lab as well. These vectors have to have a certain capacity for foreign nucleic acid. If it's low, it's not good. They can't cause disease, of course. You don't want to cure someone's genetic disease and then kill them with the vector, obviously. And you have to have a promoter, that is a sequence in the vector that makes RNA in the right tissues and cells. So here are just some examples of genetic diseases that could be treated using viral vectors, because we know for each disease what the defect is. It seems to be a single gene. So for example, hemophilia A, factor 7 deficiency, cystic fibrosis, a defective salt transporter. So in theory, you can give patients the gene, the functional gene, to restore function and take away the disease. In practice, this has been very difficult to do for a variety of reasons, but there have been a few successes. Some of the vectors that are used a lot are adenovirus vectors. These are large viruses. You can put a lot of foreign sequence in them. And in fact, you can cut out most of the viral genome from these and put in foreign sequence. And we may talk about some examples of how these are used. The other vectors that are commonly used are retrovirus vectors. These are viruses, remember, with a plus-stranded RNA genome. They encode a variety of proteins. But what you can do with retroviruses is you can put the proteins on separate plasmids and reconstitute particles. So for example, here we have 2 different plasmids. One plasmid is encoding the capsid protein, and another is encoding what we call the envelope glycoprotein. So you'll understand all of this better after a few more lectures. If we put both of these plasmids into cells, we get empty virus particles without genomes. But we can put a third plasmid in here, which, say, encodes a gene of a human that you want to correct, and now you will have retroviruses containing the foreign gene. And lentiviruses, which are a kind of retrovirus, are used for gene therapy applications as well. They infect many different cell types. And this is an example of lentiviral gene therapy to treat a neurologic disease. This is called X-linked adrenoleukodystrophy. It's caused by a defect in a gene needed for a transporter function. So you can restore the function of this gene by giving patients the gene without the mutation. So what was done in 2 patients with this gene is they took bone marrow from these individuals and put them in culture and then infected them with a lentivirus that expressed the gene that they needed. And then the cultures were allowed to expand and then put back into the patient. And in these 2 individuals, they showed regression of the disease as shown by these x-rays after a period of time. So this was actually the first very successful use of gene therapy, which just happened in November 2009. But it's an example of what you can do with viral vectors, and this all comes from being able to clone viral genomes and have them express as viruses when you put them in cells.